So we're moving on to molecular genetics, which is chapter 18. We actually skipped over chapter 17. We're going to talk about the molecules first and then how heredity actually works second. Chapter 16 and chapter 18 together make up Unit 1A. So when we're done chapter 18, that's when we'll do the first unit test. We'll be finished it this week. It won't take us an incredibly long time. And then chapter 17 will be its own whole thing. We'll do a separate one for that, and that will be the Unit 1B test. So the first two things I have up here are abbreviations, which we are going to use a lot in this chapter. Does anybody know what DNA stands for? Oh, someone's going to try. Go for it. Ooh, she knows. So it stands for deoxyribonucleic acid. Now, perhaps when you learned about heredity in previous years, people said, you don't really have to know what the letters stand for. You just have to know that DNA is the molecule that carries your genetic information. And in this case, I'm going to say that's actually not the case. It's important that you know what DNA stands for, because we're not just going to talk about DNA. We are also going to talk about RNA. RNA is a similar molecule, ribonucleic acid. Both of these molecules are important for genetics. Now, we will talk about in quite a bit of detail exactly how they function, but as an overview, we know that DNA is the molecule that is controlling everything. It is the director, it is the boss. And we know that your DNA is located in the nucleus. But the nucleus is not the only place that's involved with genetics. RNA is involved with expression and with protein synthesis. So RNA, which is a molecule quite similar to DNA, doesn't have to stay in the nucleus. It goes to other places in your cells. And because of RNA, the information in your DNA gets expressed. That means something actually happens because of it. A protein gets synthesized, and now you have a particular color of hair. So without RNA, nothing that the DNA is telling your cell to do would actually happen. The DNA is the base information, it's the boss, but without the workers, nothing will actually occur. So the ribonucleic acid leaves the nucleus. In fact, some of it is not even ever really in the nucleus of your cell. So the DNA is more or less confined to the nucleus, whereas RNA can go other places and do other things. So if you had to do an analogy here, like I was saying, the DNA is like the boss, and the RNA are the people who work for the boss. So the first thing I want to show you are just some people who are associated with figuring out that DNA and RNA are important to heredity. A few of them we're just going to mention briefly. Some of them we'll talk about a little bit more because their contribution is perhaps a little bit more detailed, or it's one that you are specifically required to know about. So I have a few people's names up here just to give you an idea of how many people it took for us to come to the understanding that we're at. A guy named Phoebus Levine was one of the first people to have isolated nucleic acids. Now, since DNA is deoxyribonucleic acid, obviously him isolating this chemical compound would be important to us studying DNA. A guy named Thomas Morgan was involved with discovering that genes are on chromosomes. And then the next two sets of people kind of are involved with the same idea. Frederick Griffith is a pretty important one. In fact, if I had to pick hey, who would they ask you about on your diploma? I would pick Frederick Griffith. Um, he did a study so that he could identify something called the transforming principle. And so the picture that's below, if you have the notes printed, can also be found in your textbook. In your textbook, it is right at the beginning of chapter 18, and I'll get you the exact page number here. It is page number 625. So Frederick Griffith goes with this picture. He figured out the transforming principle. I'm going to explain what he did first, because to understand what Avery et al. Did would be difficult without understanding this experiment first. So if you look at the picture, these are supposed to represent 4 different trials of an experiment that Frederick Griffith was doing. In the first trial, he injected mice with what we would call non-virulent cells. So he is studying how viruses can affect the basically life or death of these mice. And in the first part, he injected non-virulent stuff. That means it doesn't have the viral properties. It's not going to kill. So he would expect, if he does that, like it shows in the picture, the mouse lives at the end. Nothing bad happens to the mouse. So that's number 1. In number 2, he does something different. He injects virulent cells into the mouse, and of course, the mouse dies. So those first 2 were sort of his test trials. He wanted to make sure that what was supposed to happen happened. If I inject something that's non-virulent, you shouldn't die, and if I inject something that is virulent, you should. Then in the 3rd and the 4th, he did some other things to see if he could affect whether or not this virus was going to kill the mouse or not kill the mouse. In number 3, he took a virulent strain, so one that is supposed to kill you, but he treated it with heat. That should mean that it was destroyed. It was no longer active. Heat, as a general rule, kills things. There are not very many organisms that can live through excessive heat. So it makes sense that at the end, the mouse was okay. We had a virulent strain, but we killed it with heat, and the mouse still survived. Then we get to the 4th one, where something kind of crazy happened. He took a virulent strain that he treated with heat. So like he saw in the 3rd trial, the mouse should have been able to live. But he also injected, so you can see there's 2 different kinds of dots, the blue ones and the heat-treated ones. But he also injected non-virulent strain. And what happened? The mouse died. That's weird. When he injected the non-virulent strain, the mouse was okay. When he injected the virulent strain treated with heat, the mouse was okay. But when he mixed them together, the mouse died. That was weird. And so the explanation for this is that there was transformation. The virulent strain, even though it was not active, transformed the non-virulent strain. And we know now it's because of DNA, because DNA was present. And so basically the non-virulent strain became virulent because the DNA was present for it to happen. Now, I told you I would wait to talk about Avery. I'm actually going to talk about this. If you are reading a science article, which hopefully you will do because you're going to present one to the class at some point, when you see et al., that means and a bunch of other people. So Avery et al. Means there was a guy named Avery and a bunch of other people doing the study. It's a scientist's lazy way of saying there were lots of authors and I don't want to write all their names. So Avery and his colleagues figured out that the transforming principle that Frederick Griffith had spoke of worked only when DNA was functional. So Frederick Griffith maybe didn't know that DNA was the molecule that was making this work, but Avery et al. Figured out that this transforming principle only worked if DNA was functional. Basically, they did two tests. In one test, they destroyed proteins. In another test, they destroyed the DNA. They used enzymes to break it down. And the transforming principle worked in this case, but did not work here. So if we use an enzyme that destroys DNA, the transforming principle does not apply. And if they used an enzyme that destroyed proteins, the transforming principle still applied. Now the next two people, Hershey and Chase, Hershey is a man, his name's Albert, Chase is a woman, her name is Martha. They did some more studies to really further the idea that DNA and not something else is the molecule that gets passed on. Now there is a picture that shows something similar to this on page 626 of your textbook. Here's basically what Hershey and Chase did. They used radioactive testing, and they determined that DNA is passed on. And what they were studying, or the DNA that they were looking at, was the DNA that gets passed on from a virus to a bacteria. So they did two different tests, the one on the left where things are colored red, and the one on the right where things are colored green. Their tests were with bacteriophages. Bacteriophages are these weird structures here that look kind of like a spider thing with legs. If you've ever seen the movie Wild Wild West with Will Smith, the bad guy rides around on this machine that totally looks like a bacteriophage. But a bacteriophage is a virus that attacks bacteria. That's what it is. So they had two bacteriophages. In one of them, they attached radioactive markers to proteins. And since sort of the capsule, the outer coating of the bacteriophage is made out of protein, the whole outside of it was red. In the other trial, they labeled the phosphorus, which is part of the DNA molecule, and so what is in the sort of head of the structure, which is the DNA, was colored green. Then they allowed the phages to infect bacterial cells. So when a phage affects a bacterial cell, basically it punctures a hole in the cell membrane that allows its DNA to be inserted into the cell. So you can see here, this would be like A and that would be B. First it attaches, then its DNA goes from the phage into the bacterial cell. Then you have blending, which is basically where the DNA that the bacteria already had and the DNA that the phage inserted mix together and sort of become one set of DNA. At the end, they found, after centrifugation, has anyone ever seen a centrifuge before? It's like a big round machine, and you put little vials in it, and it spins it around really fast. You might have seen one on a TV crime show without knowing that that's actually what it was. But basically, centrifuges separate things. That's their job. So they're just mixing everything up, spinning it around really fast. What they discovered is at the end of this one, there was no sulfur in cells. So there was nothing inside of the bacteria based on the protein that the bacteriophage was made from. But on this side, the bacterial cells contained phosphorus, which was attached to the DNA. So based on their experiment, the bacteriophage, which is a virus, did not pass on its protein, which was the sort of prevailing idea. It was actually passing on its DNA. All right, so we're going to talk a bit about the structure of DNA and then who figured some of this stuff out. Now, if you have the notes printed out, you have some of these pictures, but I do want to say ahead of time, at no point will you have to have memorized the exact chemical structure of all of the parts of DNA. Generally, when we draw DNA molecules, we generalize. We talk about the pieces that it's made up of, but not every individual atom. So if any of you have already taken or are in chemistry, we are not going to draw structural diagrams of DNA molecules. There is one here just so I can show you what it's made out of. But generally we'll represent DNA a lot like this picture that's down here, with just some shapes and some letters. So DNA and RNA, for the most part, are made of the same things. They are made out of nucleotides, which are repeating units. In chemistry, we would call them monomers. Mono means one. And if you take a bunch of monomers and put them together, you get a polymer. So DNA is a polymer. Poly means many. It's a bunch of units put together in a repeating fashion. So we need to know these nucleotides, these repeating units, what are they made of? They are made of a 5-carbon sugar. Now, in the case of DNA, it's deoxyribose. In the case of RNA, it's just ribose. But that part of the word, the D or the R, that's the sugar. So I'll just note that there so that you know that that part of the name represents the sugar. Then it has a phosphate group. Now, phosphate is technically a phosphorus atom with a bunch of oxygen atoms surrounding it. We usually just represent it with just a P so that you know that it's phosphate. And we have a nitrogen base. Now, the nitrogen base obviously has nitrogen in it. That would be a distinguishing thing. And there are 4 choices for what the nitrogen base can be. So there are 4 different possibilities here. It can be A, T, C, or G. Now, those are the abbreviations. I would say that it's pretty common for us to use just the abbreviations. Of course, I'll have the actual words here in just a second. But there are 4 different bases that the nucleotide could be made up of. So if I take a look at my molecule here, just so that you can get a visual, there will be some sort of phosphate. There can be 1 or 2 or 3, and the number is not something that I want you to worry about, but phosphate, phosphorus and a bunch of oxygen, that's part of it. Then there is this part here, which is the sugar. And notice how there's kind of a pentagon shape there. That's because the sugars have 5 carbons in them, and that tends to make a pentagon shape. And then attached off to the side, there would be the base, and because there are 4 different bases, it doesn't always look the same. But those are the 3 pieces that make up a nucleotide: the phosphate, the sugar, and the base. Now over here on the right, I have a bit more information about the bases. We could split the bases into 2 categories. Category 1 would be the purines, and category 2 would be the pyrimidines. Now their actual molecular structure is shown there, and like I said before, the actual chemical structure is not a detail that generally you would have to know, but I wanted to put the structure so that I could show you this idea of how many rings they had. The purines have 2 rings. Now, we don't mean a ring like a perfect circle. In chemistry, a ring is just when something doesn't have a beginning or an end. The 2 that belong in this category are the A and the G. A stands for adenine. I'll write it a bit bigger here. And G stands for guanine. And every time I hear that, I think about the word guano, and then I have a little giggle to myself. It's not bat poop. So those are the 2 purines. They have 2 rings. Yes, they have other atoms attached to them, but it's the fact that they have 2 rings that I want you to recognize. Category 2 is the pyrimidines. Now you'll notice there are 3 things, and you're going to be like, I thought you said there were only 4 total. You're a crazy person. And that's because the 3rd one goes with RNA, not DNA. In the pyrimidines category, recognize that aside from other atomic differences, they all have 1 ring. So the pyrimidines are the C, cytosine, the T, thymine, and the U is uracil, which we'll talk about when we talk about RNA structure a little bit later. But those would all be considered pyrimidines. So the way I remember this is that the longer word is the smaller structure. Pyrimidine is a longer word, but they only have 1 ring. Purine is a shorter word, but they have 2 rings. Now the next thing is a little bit about DNA and how things are set up. Based on what we know now, DNA's basic structure is that on the sides, you have a backbone made out of alternating phosphates and sugars. So on this picture, the green circles are the phosphates, and the orangey colored pentagons are the sugars. And on both sides of a DNA molecule, those things alternate. Phosphate, sugar, phosphate, sugar, phosphate, sugar. They go in opposite order. So if I look at the other side, it starts with sugar and then goes phosphate. So they go in opposite order. This section would represent 1 nucleotide. So if I were to look at a whole DNA molecule, I would see a bunch of nucleotides down one side, and then I would see a bunch of other nucleotides down the other side. So nucleotides are these little units. Each has a phosphate, a sugar, and a base. And when I put a bunch of them together, I can make a molecule of DNA. So one of the things that you might notice if you look at pictures of DNA is that A and T always seem to go next to each other. G and C always go next to each other. This comes from, or we know about this because of Chargaff's rule. So this was a scientist, and what he discovered was the percent A that a person had would be essentially equal to the percent of T that they had. And from this, he determined that they should be present in the same proportion. The same is true for C and G. If I were to analyze how much cytosine a person had, the amount of guanine should be the same relative amount. So we're going to pause the notes here for a second and just go to your text. So on page 627, at the bottom of the page, it says exercises. And we are going to look at the two questions that are there. This would be the typical way that we would ask you about Chargaff's rule. We wouldn't ask you what the rule is. We would ask you some actual numbers. In biology, sometimes it's hard to find questions to make numeric response questions out of. Since an actual number is the answer here, this would be a good one. In question number 1, they give you 2 letters. I would say that is a really easy version of the question, and I would not expect a diploma question to give you this much information. What do I know? Well, I know that if there is 34% A, there has to be 34% T. A and T, adenine and thymine, go together. Since they told me there was 16% C, I know there should be 16% G. And if everything went well, it should add up to 100%. And you should check this and make sure, because it would be easy to say miscalculate and end up with 110%. You can't. You can only have 100%. Now that question, like I said, would be the easier version because they told you one of each pair, so you just had to know which thing it was paired up with. I think it would be a good idea for you to think of a way to remember that A and T go together and that C and G go together. I'll tell you my way, but it's probably not going to be very meaningful for you. When I was in high school, so this is how well it worked, in my tiny little 28-person class, there happened to be people who were dating each other that had those names, so it worked out fantastic. There was a boy named Adam and a girl named Tara who dated each other, and then there was a couple where their last name was Gadet and Cracknell. So we made that joke the very first day that we learned this in bio, and I have remembered it since 1997, which is when I took biology and you were not born. So if you can, think of a way that will make you remember this. Some people do the very simple thing to remember. A and T spell a word. It spells the word at, and then you just put the other letters together. But whatever works for you, you need to think of ways that are funny to you or memorable to you that will make you remember it. Now, question 2, I think, is more realistic. They give you one number, and you have to determine all of the other numbers from it. I would always start with the easiest thing. The easiest thing is for me to say, well, if there's 24% A, there has to be 24% T. Together, that accounts for 48%. So I know that the rest of it has to be 52%. That's what's left to make 100. And I know that there are 2 letters, and they have to be present in equal quantities. So I would know that C and G each have to be half of that 52%. So if I take 52 and divide it in 2, I get 26%. So that is the way I would go about solving this. Now, you are allowed to use a calculator on the diploma, although I think this is math that you should all be able to do with just your brains. I would not stop you from using a calculator, although I would encourage you to try doing this kind of stuff without one. So this is Chargaff's rule. The proportion of adenine and thymine should be equal. The proportion of cytosine and guanine should be equal. They don't have to be exactly 50% each. If you look at the first question, you would see up here this accounted for like 68%, and this was only 32%. So please don't think that they each have to be close to 50% and then they have to be equal to each other. There's no real set rule about that. The only thing that's true is that adenine and thymine should be about equal to each other. So, how did we figure out, oh, that's French too. My goodness, I was having a really good day here. We'll just get rid of that. So here we are talking about the double helix of DNA. Now we know what DNA is made out of. We know that it is the genetic molecule that's getting passed on, but we still needed people to help us figure out what DNA looks like. So there's a couple of other important people. Now, if you're lucky, the last time I was talking to people about item writing, it sounded to me like we were going to actually put some questions about people who studied DNA on the diploma exam. But I think that Rosalind Franklin is another one who you should absolutely know. You should know what she did. She took X-ray images. And since one of the things in the biology curriculum is about technology, she would be a perfect person to ask a question about because she helped us figure something out about DNA and she used a technology to do it. So she took X-ray images and we figured out some things about DNA. First of all, we figured out that it is a double helix. Now I wrote the word twice, so I'll explain what it means. I'm sure you all know what a helicopter is, and maybe you'll recognize that the word helix and helicopter have a bit of the same thing. A helicopter has blades that spin on the top of it. A double helix is the difference between a ladder that just has straight rungs down the side and a twisted ladder. Now, I am a poor artist, but if I was to try to draw a twisted ladder, I would draw, oh gosh, that's so bad. I mean, that's horrible, it's ugly. But imagine one of those kind of rope ladders. I was at this place, you should all go there, it's called the Flying Squirrel. It's like an indoor trampoline park. Oh my gosh, you'd think only kids would have fun there, but no. But they have sort of a rope ladder that you're trying to climb on and not fall into a foam pit. But you can spin a rope ladder and kind of make it twist. And that's what DNA is like. It's like a ladder that we've taken and twisted around. We figured out that the nitrogen bases are on the inside. What she actually was studying was how it reacted with water, and that helped her determine this part. And the nitrogen bases, because they're on the inside, that means the sugar and the phosphate is on the outside. Other things that she could tell from these X-ray images is that there was a constant distance between the sugars and the phosphates. So it was like every step of the ladder was equally spaced. And that would be preferable. If you've ever walked up stairs that are not equally spaced, like you get up to a landing and then the stairs change shape, you trip because your brain is used to a particular distance between stairs. So there's a constant distance. When I say constant distance, I mean like this way. Now, the other thing that we realized in doing all of this is some things about the way that the DNA molecule was attached together. And so that's what the couple of words are down at the bottom. I'll just mention what Watson and Crick did. So Franklin did these X-ray images. Watson and Crick actually modeled DNA. So she took these X-ray images and took a bunch of measurements and figured some things out. But Watson and Crick are the ones who actually came up with the model of what a molecule of DNA might look like. Now, because of their studies, because of what Franklin and Watson and Crick all did, we know that there are complementary pairs. That means A goes with T, so adenine with thymine, C goes with G, cytosine with guanine. When you look at a molecule of DNA, there are always two sides. One of the letters will be on one side, the other will be on the other side. And these are attached by hydrogen bonds. Now, if you've taken chemistry, you would have a detailed description of what a hydrogen bond is. If you haven't, basically, a hydrogen bond happens when a hydrogen atom is touching something very electronegative, and it creates this attraction between hydrogen and a neighboring molecule. So it's basically an electrostatic attraction between something positive and something negative. So if you think about the way static electricity works, it's not exactly the same thing, but it's the same sort of principle. It has to do with electron arrangements. Hydrogen bonds are intermolecular. That means they're between molecules. They're not as strong as, say, a covalent bond that holds two parts of the same molecule together. So it would be easy to break those hydrogen bonds. That's a weaker bond than, say, the other chemical bonds in DNA. The other thing we know about DNA because of their studies is that it's what we call anti-parallel. That means that the two strands go in opposite order. So like I showed you on the previous page, if one goes sugar, phosphate, the other goes phosphate, sugar, phosphate, sugar. Chemically speaking, you'll see some numbers. And so I'll just show you what they are so that they don't freak you out. Sometimes you'll see a 5 with a little line. If I were to read that out loud, I would say 5 prime. Sometimes you'll see a 3 with a little line. If I were to read that out loud, I would say 3 prime. When we're talking about chemical molecules, we would talk about their orientation and the way that they're facing. And that's all these numbers are really telling you, is which way the molecules are facing. If you look at one side, the 5 is at the top and the 3 is at the bottom. If you look at the other side, the 5 is at the bottom and the 3 is at the top. The numbers themselves are not a main part. You'll see them because it's always how we talk about DNA. The main idea is that the strands are flipped from each other. One of them goes one way, the other one goes the opposite way. Does anyone have any questions that they would like to ask about that? So that's a bunch of information about DNA. And what you'll see is all of the studies are us talking about DNA, even though RNA is a really similar molecule. RNA studies usually piggyback on DNA information that we already have. So I'm not going to show you any more studies right now, but just a little bit of information about RNA. So like I said at the beginning, RNA is similar to DNA. There are 3 differences from a molecule of DNA to a molecule of RNA. The first difference, the sugar is ribose, not deoxyribose like it was for DNA. The second difference, like I mentioned earlier, instead of thymine as a nitrogen base, you have uracil. And the third difference is that RNA is always a single strand. So it's a molecule that's a lot like DNA. It has the same basic setup except for those 3 things. So if I were to look at RNA, which is what this picture is showing, there's no other side. There's only one strand. It still has a phosphate. It still has a sugar. It's just that the sugar isn't exactly the same. It still has adenine. It still has guanine. It still has cytosine. But uracil is different. Other than that, they're made of the same thing.